Well, thank you for joining us today and welcome to the first session of Agrigenomics Track on day 2 of the APJ Genetic Solution Tour 2020. My name is Ryan Harvey and I am your moderator for this session. In this session, we will hear about the Ion Tolerant AmpliSeq targeted genotyping by sequencing platform and its use for agrigenomics applications. Just a note, if you have any questions that arise during the following presentations, we encourage you to submit them in the questions and answers box, and our speakers will address your questions following the presentations. Firstly, today we will hear from Dr. Takeshima, who is a researcher at the NARO Institute for Crop Science in Japan. Today he will be talking to us about how the Ion Tolerant AmpliSeq platform, a predecessor to the AgriSeq platform, was used in his laboratory for the identification of genetic loci associated with self-compatibility, pre-harvest sprouting, and maturity time. Welcome, Dr. Takeshima.
はい、皆さんこんにちは。農研機構次世代作物開発センターの竹島です。今回はジェネティックソリューションツアーの発表の機会をいただきありがとうございます。ウェブセミナーは初めてですけど、非常に楽しみにしています。本日は普通そばという作物についてイオントレントの提供するアンプリセックテクノロジーを用いて効率的に育種研究を進めるという私たちの一つの手法をご紹介します。今回普通そばのようなゲノムのデータベースがまだ十分に整備されていないマイナークロップでいかに効率的に遺伝解析を行うかといったテーマです。今回解析の例に挙げます標的の形質は普通そばの自家不和合性、農業上重要な形質である穂発芽耐性また冬熟期です。これらの形質についてアンプリセックの実験系の開発と成果の概要というものをご紹介します。それでは発表に移っていきます。まず本日お話しします内容です。はじめに普通そばとその特徴的な生殖様式である異形花形自家不和合性の紹介をします。また普通そばのゲノムのデータベースの状況や遺伝解析の進捗なども紹介いたします。次にその状況を踏まえて私たちがどのような戦略で遺伝解析を現在行っているかというご紹介をいたします。ここで自家不和合性や穂発芽耐性についての結果の概要とともに紹介していきます。また最後に私たちの最近の他の仕事なども少し紹介できればと思います。はい、それでは普通そばの紹介です。普通そばはタデ科そば属の一年草です。植物としての特徴として自家不和合性に起因する多食性の植物です。また栽培上の特性として乾燥や低温に強い耐性を持ちます。また非常に強いアルミニウム耐性を持っており酸性土壌でも適応性があります。さらに播種から収穫までが80日程度と生育期間が非常に短いというのも特徴の一つです。このような植物上の特徴を持っています。普通そばの利用にしましては一般的には種子を調理して利用します。地域によって調理法が分かれておりましてロシアや東のヨーロッパの方ではこの史実の皮を剥いた状態でそのまま炊き込んでおかゆにして利用することが一般的です。カーシャーという食べ物になります。一方、アジアやヨーロッパではこの史実を粉にして加工してから調理します。左下の写真はフランスなどで食べられているそば粉のクレープ状のガレットというものです。この写真はフランスのガレットではなく先週私が自作したガレットですが、味は良かったです。皆さんガレットで Google してもらうともっと綺麗な正式版が見えますので、検索してみてください。一方右下の麺のようなものはアジア特に日本でよく食べられます。日本ではそばの麺は伝統的な食品として親しまれていまして、大晦日では日本人がみんな 斉にそばを食べるという楽しいイベントがあります。でこれらのそばですが、食物としての栄養学的には、ビタミン B 群や良質なタンパク質を豊富に含んでいます。またルチンという機能性の成分も含みます。さらにグルテンフリーということで、最近注目されている植物の一つでもあります。このようにそばはマイナーながらでも世界でたくさん食べられておりまして、生産も世界の各地でされています。こちらがそばの生産国トップ10の、国になります。で FAO の2016年のデータから取っています。左からロシア、中国、ウクライナ、フランス、ポーランド、カザフスタン、アメリカ、ブラジル、リトアニア、日本となります。このようにそばはロシアのような亜寒帯の気候区分の地域からブラジルのような熱帯の気候区分の地域まで非常に幅広いところで栽培されています。これはそばの持つ適応性の高さを示しています。以上のようにそばは、適応性も高く収穫時間も短い、さらに栄養価も高いという作物ですが、まだまだマイナーで作物として遺伝的な改良がされていません。でどのように遺伝的な解明がまだ改良がされていないかということを紹介いたします。まずそばはまだ脱粒性が少し残っています。この左上の写真のように、子実と植物体を結ぶ柄の部分、加工の部分が細く、収穫時期にボロボロ脱粒するという問題が未だに残っています。また右の写真のように、品質に大きく影響する穂発芽という現象もまだ解消されていません。この他に、例えばそばには非常に強いアレルギー物質があります。これまでの研究でそばの主要なアレルゲンとして3つファゴ E1、ファゴ E2、ファゴ E3 という物質が同定されていますが、この改良には至っていません。さらに熟期のばらつきも問題になっています。右上の穂発芽の写真のように黒く熟した実の隣にまだ青く未熟な状態の種があります。一株の植物体の同じ位置の花でもこのように熟期がばらつくので、畑の中の集団で見るとさらに熟期のばらつきというのがあります。でこれが何が問題かといいますと、生産者の人たちは畑の熟し具合を見て収穫期を決めるので、全ての植物が熟すまで待っていくと、ボロボロ先に熟したものから脱粒してしまうということで、この熟期のズレというものも脱粒を助長する一つの原因となっていますが、これもまだ改良されていません。またそばは乾燥や低温に非常に強いですが、過剰な水分に非常に弱く湿害が大きな問題となります。特に日本のような湿潤な地域でそばを育てるとなると湿害が大きな問題になりますが、その耐性というものもまだ改良されていません。以上のように、例えば脱粒や穂発芽など他の作物ではまず改良されているような、形質というのがそばではまだ残っていて、改良の余地がたくさんあるというふうになっています。現状でも。でなぜそばの改良がここまで遅れているかという理由の一つとしてそばの特徴的な生殖様式というものが挙げられます。でそばの生殖様式というものは異形花形自家不和合性というものです。でこの自家不和合性のメカニズムは油中などの不和合性のメカニズムとは全く異なる方式をとっています。で異形花形自家不和合性のルールとしては、異なる花のタイプの花粉でしか受精ができないということです。でそばでは二形花というタイプを持っていて、普通のそばの集団の中では2つのタイプの花のタイプしかありません。で1つの植物体には1つの株、1つのタイプの花しかつきません。で左側の中東が長く脈が短いというピンの花で、右側の中東が短く脈が高いスラムの花があります。ピンの花粉がスラムの中東につくと受精はしますが、ピンの花粉が同じピンの中東につくと受精はしません。同様にスラムでも異なる花のタイプの中東につくと受精はしますが、同じ花のタイプでは受精はしません。 で一つの植物体については一つのタイプの花しか咲かないので、同じ自分の花のタイプが同じなので受精ができないという不和合性のシステムになります。で他の不和合性のシステムと違うところは、個体が違っても花形が同じだと受精ができないというタイプで不和合性になっていますので、他の不和合性より強い不和合性のシステムを持っています。このような生殖様式を持ちますと、優良な個体が現れてもその形質を次世代に固定できないや自植ができないので、一般的な遺伝解析がしにくいという現状があります。でこの問題を解決するために、我々はまず自植できるそば系統を作ろうということをこれまでやってきました。その手法としましては、多食性である普通そばに、その近縁の野生種で自植の形質を持つファゴヒラムフォモトロピカムという系統を交配して胚培養でレスキューさせて主幹雑種を作るという手法で自植性の普通そばをまず開発しました。でこの自植性の普通そばを用いることで遺伝解析 F2 やリルでの解析ができるという準備が整いました。また遺伝解析にはゲノムのデータベースが非常にあることが有効なんですが、最近までそばのゲノムのデータベースはありませんでしたが、3年前に私たちと同じ研究グループでやっていただいている京都大学からそばのゲノムのデータベースがリリースされました。でまだドラフトゲノムの状態です。そばのゲノムは染色体が8本2倍体です。で推定ゲノムサイズが 1.2 ギガでスカフォールドが38万7000に分断されているようなドラフトゲノムが公開されました。でこのような遺伝解析ができる材料とゲノムのデータベースが揃いましたので、最近ようやく私たちは普通そばにおいて遺伝解析ができるという状態になりまして、この現状から一気に解析を進めていこうというふうな戦略をとっています。で次のページから、私たちの、実際普通そばにおける遺伝解析の結果を報告していきます。で解析の対象とする形質は、自家不和合性、穂発芽、等熟期です。でこちらが、標的の形質です。で解析のストラテジーとして、次世代シーケンサーベースのバルクルセグレガントアナリシス、QTL-SEC と、ターゲットアンプリコンシークエンス、アンプリセックを合わせた、解析手法で、効率的に対象形質の原因遺伝子差を見つけようという手法を取りました。まず QTL-SEC の説明だけ先に簡単にします。皆さんご存知の方も多いかと思います。2013年高木らによってプラントジャーナルで報告された量的形質に関わる遺伝子差を効率的に次世代シークエンサーベースで検出するという手法です。具体的な概要を紹介しますと、草丈について分離する F2 集団 F2 以降の集団に関して、草丈が低い集団のバルクの DNA と、高い集団のバルクの DNA それぞれを、ディープシークエンシングしていきます。そこで、草丈の高い集団に特徴的なスニップ、低い集団に特徴的なスニップというのを検出させて、スニップインデックスを計算し、そのスニップインデックスが高いスニップが座乗する遺伝子の領域に、草丈に関わる遺伝子があるんじゃないかというような、推定ができる非常にスマートな手法です。ただしこの解析手法はそばではまだ使えません。で理由として、この解析は物理地図に落とし込むことが必要だからです。ただそばのゲノムの情報はドラフトゲノムでスカフォールドが38万7000に分断されています。 N50 は25キロです。つまり染色体8本で8枚の絵が描けるべきものがジグソーパズルのピースが38万個に分断されているということです。でそれぞれのピースがどこに入るかもわからないということです。この状態で QTL-SEC しますと、例えば形質に非常に高いインデックスを持つスカフォールドが A、B というものが出てきても、一つの遺伝子座に起因するスカフォールドであれば当然連続した値なので1個の原因遺伝子ということになりますが、このように違う領域に QTL が存在する場合もあります。で 今のこのそばの現状だとそれぞれがどのような遺伝子差に存在するかというのがわからないので、この状態のままでは QTL-SEC は扱えないということです。でここを我々は改良しようとして、アンプリセックを組み合わせました。でストラテジーとしては非常に単純で、物理地図じゃなくて連鎖地図上にスカフォールドを落とし込もうという手法です。でそれを一度にやってしまおうという解析です。で次にマーカーのデベロップメントのストラテジーを紹介します。まず、解析の集団の紹介をします。集団の親として多色性で穂発芽が強い KY29 という親、また、自食性で、穂発芽耐性が弱い KSC7 という親を作りました。この F2 集団では、多色と自食の形質で、穂発芽の強い弱いの形質が分離します。まずこの2つの親の、ホールゲノムシークエンスを読みました。次にこの F2 の集団に対して、自食のバルク、多色のバルクに関して、ディープシークエンシングをしてそれぞれ、100個のスニップインデックスが高いスカフォールド領域を検出しました。また同時に同じ集団で、穂発芽耐性が高いバルク、低いバルクそれぞれ合わせて100個のスカフォールドを検出して、それぞれマーカーを作っていきました。でこの100個のマーカーをそれぞれだけでやってしまうと、先ほど示したような、どこに存在するかわからない領域というふうになってしまうので、これにプラス合わせて300個のゲノム全体のスニップスを検出できることが予想されるホールゲノムのマーカーも作りました。そして合計で500のプライマーペアがセットになったカスタムパネルというものを作りまして、これで解析をしていくということになりました。で今回3つの独立した集団で解析しましたが、代表的な1つの集団だけご紹介します。まず F2 での穂発芽の分離になります。で穂発芽の耐性が強い KY29 と穂発芽の耐性が弱い KSC7 の F2 の集団です。左に行くほど穂発芽が強いことになります。でこの分離の様式から見ると KY29 の持つ穂発芽耐性の遺伝子座は、優性遺伝で分離の様式からおそらく2遺伝子支配ということが予想されます。でこの集団の高い領域のバルク、低い、集団のバルクをそれぞれ、ディープシックして先ほどのアンプリセックのマーカーを作っています。次に、自食多色の形質に関してです。この自食多色の形質というのは、S 遺伝子だという一つのシングルローカスで、支配されていることが分かっています。ただその遺伝子の物理的な位置ということはまだ分かっていません。で今回の F2 の集団において、自食が多色に対して優勢なので F2 ではだいたい1対3の割合で多色自食が出てきます。でそのような分離比に分かれてくれたので、このまま自食のバルク、多色のバルクでまたディープシークエンシングをして先ほどの100個のマーカーを作っていきました。で今回先ほどお見せした100個の自食多色にリンクしたマーカー、100個の穂発芽耐性にリンクしたマーカー、300個のホールゲノムのリンクしたマーカーの解析の予想として100個のこの自食多色のマーカーはシングルローカスに落ちてくれるんじゃないか。で100個の穂発芽耐性のマーカーは2つのメジャーなローカスに落ちてくれるんじゃないかというふうに予想しています。でこの300個のホールゲノムのマーカーで8本の連鎖地図が作れれば OK ということです。で解析を行いました。で解析は 4S5 のアンプリセック540のチップで行っています。で概要だけ、お示します。まず300のホールゲノムのマーカーで8本の連鎖地図はできました。でこれは SI、SC、自食多色に連鎖するマーカーの解析結果になります。この緑色で示したものが自食多色が連鎖することが予想される QTL-SEC で作ったマーカーたちです。で黄色のマーカーが自食多色の形態マーカーです。で、今100個のマーカーのうち12個しか連鎖地図には載っていませんが、同じ遺伝子型を取ったものは重複マーカーとして外されています。すべての100個のマーカーのうちジェノタイプできたマーカーはこの連鎖群1の、形態マーカーの近傍のところに落ちてきました。ですので狙い通り1つの遺伝子差を集中的に解析することができたということです。 で今回は3つの独立した、集団で行いました。でそうしますと、それぞれ、この形態マーカーを挟むような、マーカーが、できまして、それぞれの、連鎖関係というものを分かってきました。でこの中で、スカフォールド、座乗するものや ORF みたいなのも予想することができました。次に歩発芽体制に関してです。今回、歩発芽体制の100個のマーカーは4つの連鎖群に分かれました。その中でもこのグループ6の連鎖群に非常に多くの歩発芽マーカー、歩発芽連鎖マーカーが座乗していました。で同じように重複したマーカーはオミットされています。で実際 QTL 解析をしますと、最も、歩発芽体制にリンクするマーカーが座乗した連鎖群に QTL の最も高いピークも来ました。ですので、こちらも今回の QTL セックで作ったアンプリセックのマーカーで、しっかり遺伝子差の領域が絞り込めたということになります。簡単ですが以上の結果をまとめます。私たちのマーカーのシステムにおいてはまだゲノムの配列が染色体上に載っていないようなゲノムデータベースが未整備なマイナークラップにおいても一度の解析で QTL セックによる遺伝子差領域の測定や連鎖地図の構築というものがアンプリセックのデザインでできるということが分かりました。でこのようなものを使っていけばゲノムの整備というものを待たずにどんどん狙った形質の領域っていうのを狭めていけるんじゃないかということが分かりました。で他の仕事としては私たちはその自家不和合性に関わる遺伝子を違う手法でも探っています。これ昨年のパブリッシュされたものなんですが、これはピンとスラムの花形特異的なプロテオム解析からある1個の遺伝子を取ってきたという遺伝子ですという実験です。こちらも興味があれば連絡をください。以上になります。これらの研究は農研機構の研究費および JSPS の科研費の一部を使って解析しました。でご清聴ありがとうございました。何か質問や興味がありましたら連絡をください。以上で発表を終わります。
Great. Thank you very much for the excellent talk, Dr. Takeshima. We will now be moving into the live Q&A section here, and let's have a look here. So there's a couple questions coming through now. And the first one, Dr. Takeshima, is, are there any plants that show heteromorphic self-incompatibility properties?
So thank you for question. So Primula, Turner, and Linux are showing heteromorphic self-incompatibility property. Thank you.
Thank you. We have another question here as well. Is this the most common strategy to drop scaffold onto linkage maps but not onto physical maps?
So I think it's commonly used technique to create the pseudomolecules. So when converting a draft genome into a pseudomolecule, I think there is a method to drop each scaffold on the linkage map and look at the linkage relationship to create a chromosome-based genome database. So, however, in this case, it seems that there is no report on the method of dropping scaffolds on the linkage map by performing QTL-seq on the draft genome that has been finally debated like this time. Thank you.
Thank you. We have another question here as well. On the slide titled as Distribution of PHS Tolerance Cross A, you explained that the locus of pre-harvest sprouting is probably dominated by two major genes from the pattern of distribution. How did you find that out?
Yeah, it is expected at a glance that it can be applied to two dominant gene segregation patterns. So I mean the ratio of the 15 to 1. So for example, if more polygenes are involved, it will take Gauss distribution with a peak around the center of the parent. So on the other hand, if monogenic locus is dominant, so the ratio is 3 to 1, the number of individuals will be higher in the part that is easy to show pre-harvest sprouting. Thank you.
Thank you. We have another question here as well. Do you think that this methodology is efficient for this species, which does have a draft genome but does not have a physical map?
Yes, I think so.
Thank you. And a couple more questions here as well. Are the 300 markers for linkage map used not for genome-wide linkage map, but for connecting the 100 selected marker segments?
So 300 markers are used to create a linkage map of the whole genome and to analyze in which region 100 selected markers are gathered on the linkage map. So if we do not do this method, we will not know if the 100 selected markers are clustered at one locus or are scattered in the multiple regions. Thank you.
Thanks. And another question here as well. Are there any other conditions needed for applying this method?
So I think there will be no problem if we can properly select a genome-wide marker that can construct all linkage maps. Thank you.
Great. Thank you very much for that, Dr. Takeshima. We'll just wait and see if there's any other questions that come through in the next minute or two. Otherwise, thank you very, very much for that wonderful presentation.
Yeah, thank you, sir.
Great. So next we will hear from Dr. Srinivas Chadaram, who is our Global Product Manager in our Agrigenomics business. Srinivas is based out of Austin, Texas, and he's responsible for the portfolio of products supporting agrigenomics, including AmpliSeq genotyping by sequencing, CE sequencing, as well as working with the qPCR and microarray teams supporting agrigenomic solutions. Today, Srinivas will be talking about how the AmpliSeq targeted GBS is successfully being used as a high-throughput, customizable, and cost-effective genotyping solution in aquaculture, animal, plant breeding studies, as well as parentage testing and genetic purity. Welcome, Srinivas.
Hello, everybody. Thanks for joining us on the Genetic Solutions Store. Today, inspired by the research efforts of several scientists like Dr. Takeshima, who have used the Ion AmpliSeq technology to discover genetic loci in plants and animals. The Thermo Fisher Scientific agribusiness R&D team developed the AgriSeq targeted GBS technology using the same ion tolerant next generation sequencing platform and the highly efficient AmpliSeq chemistry for targeted agricultural applications. Today, I'm going to talk about the AgriSeq targeted GBS platform and how it has become a fast and affordable genotyping by sequencing solution for breeding programs in plants, animals, and aquaculture. Next generation sequencing is actually a very innovative and informative technology. However, it is very complex and it requires technical expertise and highly qualified trained scientists to be able to handle the complex workflows, especially in identifying the specific markers of their interest and how do you design a targeted panel, which is very highly specific and sensitive, given all the genomic complications across various species. I mean, plants have a very complex genome. Similarly, each and every animal species has their own genomic complexities. And one really needs to have the expertise to understand and navigate through all of that. And on top of that, the sequencing technology gives out you a deluge of data. You really need help in understanding the various pipelines to make sense of the data. You need bioinformatician support to really help you sieve through all that. I mean, sometimes you get megabytes of data per sample and gigabytes of data per run. And to really make sense of all of this, you really need trained personnel. And then, of course, the industry has multiple data formats that you need to work through and understand. So these are several challenges that, especially for an agriculture industry, it's becoming very daunting to apply the next generation sequencing platform. So Thermo Fisher Scientific has always been a technology enabler, providing the building blocks. We have been serving the agri industry for the past several decades. And depending on what the customer needs and where they are in their genomic journey, whether are they doing early discovery phase after whole genome sequencing, looking at genome-wide association studies and doing some genomic selections and predictions, where they are looking at millions and hundreds and thousands of markers, we have the microarray platform that helps them with those applications. Similarly, if they are developing new markers and new loci that they are really researching and coming up with using our PCR or our Sanger sequencing platforms, whether for seed QCing or looking at microsatellite markers for parentage verifications in animals, we have all those platforms. But now, as the customer is trying to add more relevant markers or make it get you more genetic value into their breeding program, or likewise, you're trying to streamline and optimize which markers are really essential for you and trying to develop a known target list that is essential for your marker-assisted selection or breeding program, we have utilized the targeted GBS platform, developed that platform to help with this mid-density range, which provides you a lot of flexibility and value. And in fact, you can use our AmpliSeq chemistry, which is very efficient for multiplexing several thousand markers. It could be as small as 50 markers all the way up to 5,000 or even up to 10,000 markers. All of this reaction is performed in one single droplet reaction. And that's the power of the technology that the AgriSeq targeted GBS brings forward for our customers, especially focusing on marker-assisted selection and breeding in plants, animals, aquaculture. And Thermo Fisher Scientific has been providing these solutions across the continuum. In fact, we believe in providing a complete solution. We have several sample prep solutions that are specific to plants versus animals or aquaculture. We could always bring in those and augment our AgriSeq technology, where the genotyping solution starts with designing custom panels, followed by using the modified, reformatted AmpliSeq chemistry to suit the high agriculture throughput and scalability using barcodes, and then the sequencing, the ion tolerant sequencing platform, and then we cap it with the data analysis tools that gives you a complete solution for your needs. In terms of the workflow, it is a very simplified workflow. So for the agri customers to adapt, it starts with first designing your panel, and our bioinformatics design team provides a great service in working with you in developing high performing panels. So using those panels, and after you have isolated your DNA using one of our sample prep solutions, you start the library construction process using that panel along with the AmpliSeq library kit and the barcodes. So you amplify your targets and then barcode them. And once the barcoded libraries are normalized, then they are loaded onto the templating platform. The whole library construction takes about 6 to 7 hours. And if you are doing a 96-well protocol, you can do it manually. It includes about 3 hours of actual manual work in the 6 to 7 hours. And it is very automatable, and you could actually increase your throughput by implementing a 384-well format. And the hands-on time then becomes just 1 hour. Once your libraries are barcoded and normalized, then you are adding that onto the chip on an ion chef, where the templating happens overnight. It is fully automated. You can load all the reagents and the chip and the libraries and go home. And the next day morning, you come in and take out the chip and load onto the sequencer for sequencing. Irrespective of how many samples and how large your panel is, the sequencing on the ion tolerant sequencer only takes 2.5 hours. It's all automated. Your hands-on time is only 15 minutes to turn the system on and to load the chip, and then to turn it off and do the washes. So it's very automated. And then it takes about 6 hours for the raw data to be converted, and then for the complete analysis to happen, it takes anywhere between 6 to 24 hours. So your entire workflow from sample to data and results is about 3 days. And it is very highly scalable because you can totally automate it, and you can reduce the hands-on time and reduce the errors and increase the efficiency. And the whole system is really very nicely integrated on our Ion Gene Studio system. You have the ion torrent server, which actually is where all the data is stored. The torrent suite software helps you plan your entire run right from the beginning of labeling your samples and assigning the barcodes and planning your runs. And all the primary data is stored and analyzed here, where the electrical signals, the raw signals, are converted to base calls, and the alignments and base alignments are made. Once all of this is done, then the variant caller plugin is used and the coverage plugin is used. They make the call for the presence or absence of the variant. And all that data is further analyzed and summarized for you using the Agrigenomics toolkit, and it provides you actionable genotypes. So this entire need of trying to go back and look at raw data is taken away, because the Agrigenomics toolkit really summarizes everything for you and gives you the actionable genotypes in a format that you can use. So the interpretation, what does the Agrigenomics toolkit do and how it makes the results really easy, is that it summarizes the entire run for you, showing you what types of markers were involved in the panel, whether they're SNPs, deletions, insertions, how many amplicons were there, and what's the mean coverage and the call rate. It gives you the marker performance, and it can be categorized into into several bins as per your requirement. And similarly for the sample call rates. And we use the simple traffic signal lights, green being good, yellow or orange is be watchful, and red is that things have failed, go check out what has happened. The Agrigenomics toolkit also presents this data into very easy interpretable format in bar graph. So on this bar graph, on the y-axis, you have the number of samples, and on the x-axis, you have the call rate range. And when you look at a graph like this, where you are seeing green bars, which is good, and wherever you are seeing red bars, that means it is bad, and these samples have failed or need to be rerun. So you get an upfront idea how your overall panel has performed, how your overall experiment has performed, and you can decide on what the next steps are. It also gives you additional graphs where you can do a quick comparison. Why has my sample failed? So it will show you the mean depth or coverage of the run and how the call rates are, so you can quickly make those associations and decide what needs to be performed. So it kind of really helps you to do a very easy overview upfront. And then it provides you the capability of downloading the data in a format that you are able to do downstream analysis. We normally give the genotype metrics, which is the reference versus variant calls for all the samples. And then we also give you a top-bottom format, which most microarray customers are used to, or the parentage format, which is recommended by associations like ISAG. The Ion Gene Studio system is really scalable and flexible. The flexibility comes in with the technology. It's the same sequencers, whether it is our base model, the S5 and the S Plus, or the S Prime, which is the high powerful system with an integrated server along with it. And it's the same system that can be used for multiple applications, though here we are concentrating on plant and animal breeding application. And the same sequencer is used, and depending on your throughput, you can always change your throughput using a different type of chip. If you have a smaller number of samples that you want to quickly test, you can use a smaller chip, like say, for example, which gives you between 3 to 5 million reads. You can test about 125 plus markers for over or up to 400 samples. Or you can use a large 550 chip, which is giving you the capacity to test up to 130 million data points, where you can test anywhere between 800 to 5,000 markers for about 260 to 1,536 samples. So the system is designed such a way that it easily provides you the capability to scale, and it's also very flexible. So overall, the AgriSum targeted GBS platform is very flexible. It allows you to screen for multiple markers, SNPs, multinucleotide polymorphisms, indels, and various structural variants. You can do multi-species panels. You can run at the same time multiple species. You can it is a very powerful and scalable platform because you can totally automate it. You can run 1,536 samples in one time. You can generate several novel genotypes, and you can also have the flexibility of adding or dropping markers and editing your panels. So what all of this does is results in a very cost-effective platform because it is flexible and powerful. You're able to multiplex for several hundreds to thousands of markers in one single reaction. You're able to perform high throughput, 1,536 samples per chip, or 3,000 samples in a day on one single instrument. And if you want to increase the throughput, you can always apply a modular approach, adding more instruments. So here's some data that will help you validate our claim. So we have developed a small panel for bovine. This is the markers recommended by ISAC, a 200 SNP panel. And we've also developed, on the recommendations of several KOLs, a 1,500 SNP panel for porcine, for swine. And in both cases, and here that is what we are showing, that irrespective of the size of panels, the performance is really good. I mean, the panels will have an average call rate of 98.5 and 96.3%. They are really good performing panels. And then we've also checked for intra-run and inter-run concordance by running the same samples on the same chips and also between chips. And in both cases, for both panels, the concordance is really high, way above the 99.9% in both cases. Not only that, we've actually done orthogonal comparison by taking the help of one of our customers who had access to the Illumina bead chip. So we tested the same bovine samples on both platforms and saw that there's 99.4% concordance between both of them. And so you can really trust this technology works, and it works really well in comparison to other technologies on the market as well. So here's another example that in collaboration with one of our partners, Eurofin Biodiagnostics, we developed a sunflower 768 SNP panel where we used samples from the US grain database. They had known samples. And so we created this panel of SNP markers, which is available, and it was tested. So we ran that in-house in the Thermo lab, and Eurofins ran it in their lab. And then we also tested it with the historical Illumina array data that the US grain folks had. And so you can see there was very high concordance between all these comparisons that we have done, all in the range of 95% or 99% and above. So this technology really works, and it offers you a pretty good solution. Here's another test that we had done for the USDA, where they wanted us to screen the 19 beef-producing bovine breeds, and we did parentage analysis on them using the bovine 200 marker SNP panel, and there again, the average call rate was 98.5%, so the panel is a very well-performing panel. And in fact, last year, ISAC approved all of these markers into an expanded panel of 200 markers. And so this is readily available on the market for the livestock customers. So overall, our team has been enabling the agriculture research. You can see our timelines. We have increased the sample throughput, thereby driving the sequence costs down from 384 all the way to 1536 barcodes. It actually lowers your sequencing costs by a multiple fold. We have also developed various tools, starting with the Torrent Suite software, which is the core technology, core tool that helps with all the analysis. And now last year, we've launched the AgriSum toolkit, which aggregates all this data and presents it in a very usable and interpretable format. And we are launching an improvement later this year. Similarly, we have designed several off-the-shelf panels, which are readily available for you to order and use, which is the ISAC bovine panel. And then we've also developed a canine and feline parentage panel, which includes all the ISAC recommended markers. And we also have an additional panel for canine and feline defects. Over the years, our designs has really improved and has been able to add more and more design for more and more complex markers, starting with SNPs. We have added pipelines and designs for multinucleotide polymorphisms, indels, and we are developing pipelines for structural variants. Overall, we have always been supporting custom panels because, as you know, every genome for every species is different and it has its own complexities. And so are our customers. Our customers have a variety of interests. People are looking for genetic loci for improving their production values, and somebody is really looking at improving the disease resistance and meat quality and milk quality. Or in plants, they are looking at faster yields, higher yields, disease resistance. So every customer comes with their own set of interests and requirements, and we are able to incorporate all of that in our custom panels. So here's a comment from one of our early KOLs who have adapted. These were the Department of Fisheries, Dr. Terry Beacom. He has adopted our platform very early on in 2017. And at a talk, he shared that we had helped them in developing a panel for screening eucalyptus salmons. So our design team was able to design an assay which included 500 plus amplicons, and they could run 756 samples at a time on a single chip, giving them 95 million reads. And he praised our team really well, and he says that this was a great design because he has seen several of his American colleagues really struggle to get such a panel going on an Illumina or a competitor platform. So our pioneers are really happy using our platform. So we in Thermo Fisher Scientific believe in continuous improvement. We really work with our customers. In the past 3 years, we have developed over 150 custom panel designs covering over 40 different species. You can see all the plants and the animals and aqua species that are listed out there, and we are still counting. Every year, we get several projects where people come to us bringing in their requirements. So we continuously believe in improving our designs. We work with our customers in meeting their requirements and make all the improvements needed. We work as partners. We really believe in working together, and we are looking forward to work more and looking forward to develop such partnerships and collaborations in the Southeast Asia region. So you can check our customer testimonials going to our website at thermofisher.com, Agrigenomics. There are several videos that will provide more insights. And we have been empowering worldwide agriculture with various partners throughout this past 4 years that we have launched the AmpliSeq targeted GBS, which is a very powerful, flexible, and cost-effective platform. I thank all our team members, and now I am open to taking your questions and would be happy to answer and looking forward to developing some partnerships in the near future. Thank you.
Thank you very much for the wonderful talk, Srinivas. We have a couple of questions coming through here, and one of the first ones is, how is AgriSeq unique from other genotyping by sequencing solutions?
So AgriSeq is an amplicon-based targeted system, so whereby you are developing panels for the targets of your interest. Thereby, you will see very high consistency and accuracy. That's not the case when you, say, try a RAD-seq method, which is more random and a restriction enzyme-based platform. So that's the uniqueness of AgriSeq. Because you're using a targeted amplicon-based system, your consistency and accuracy of performance is really high.
Thank you. And another question we have here, what is the key difference between AmpliSeq and AgriSeq?
Great question. As I mentioned in the presentation, our focus was how Dr. Takeshima and others have used the Ion AmpliSeq chemistry as a basis tool. However, when it comes to production customers, both in livestock and plant breeding, they really need high throughput performance. And for that, the Ion AmpliSeq chemistry is more targeted where you are doing a research-based approach with lower number of samples. Here at AgriSeq, we were able to scale it up by modifying the workflow protocols so that you are able to run more number of samples and you are able to generate more data. So there is a difference in some of the formulation, which allows you to scale up the number of samples and generate more data as needed by production customers, both in livestock, aquaculture, and plant breeding companies.
Great, thank you. Another question we have here is, how many markers can be included on an AgriSeq panel?
So technically, the AmpliSeq chemistry, you could include up to 23,000 markers, but that's not economically viable. So as we presented that AgriSeq is a very good mid-density platform, you can target anywhere between 50 to 5,000 markers of your interest. And that's what we are seeing, that the market, after having done all the research with microarray platforms and after doing whole genome sequencing, they would like to pick and choose relevant markers for the breeding, and the mid-density is the right range for using the AmpliSeq platform.
Great, thank you. Another question we have here is, can we genotype multiple marker types in multiple species?
Yes. So the AmpliSeq chemistry provides you the flexibility to be able to design primers for multiple markers in the same panel. You could target SNPs, you could target insertions and deletions, or multinucleotide polymorphisms, or structural variants. You can design primers for all of them and still maintain compatibility that these primers are specific and sensitive to the targets that you're trying to do, because it's one single reaction where you're performing a ultra multiplex amplification reaction, right? And then when you're trying to do multiple species, you should make sure that you are amplifying or making your libraries individually for each different species. Once all the target libraries are amplified, they're all barcoded, cleaned up, and normalized, that is when you can pool all of them and run all the species together. Like, say, if you're a service lab, you may have bovine samples, equine samples, or you may have some corn and soy samples. So you prepare individual libraries with their respective panels, then combine all of them into one and load it onto your chip, and then you can sequence them together. Thereby, you are optimizing your sequencing capability and optimizing on your effective costs.
Great, thank you for that. A couple more questions we have coming through. Can we use markers or SNP information from microarrays for targeted genotyping by sequencing panels as well?
Yes, our design pipelines are created such a way that it helps in customers bringing data from other formats, most likely with microarrays, because people would have done the genome-wide association studies and have done the genomic selection studies where they have identified the markers. So you have readily available information for your marker. So that can easily be used for designing the primer panels for the targeted GBS AgriSeq platform. However, what we recommend to our customers is, in addition to those 30 or 40 or 50 bases that were used in the microarray, we would recommend that they increase it to at least 100 bases, provide us sequences covering on both ends of the marker with about 100 bases, and so it allows you to create better panels, better sensitive panels. So that's our recommendation. Ryan, any more questions?
Sorry, we have another question here. Do we need to buy additional software for running the AgriSeq GBS on the Ion Gene Studio sequences?
No. So as again in the presentation I showed, the Ion Torrent platform runs or is integrated with the Torrent Suite software suite. It is an open source tool that actually comes along with our platform, and all the plugins are developed on open source. So all of this is available and it comes with the system. And we've also added the AgriSum toolkit, which is again developed with open source tools available out there. And all of this is integrated and comes along with it. You don't have to pay any additional charges for using the software.
Great, thank you. And that probably leaves us at the end of our time here. I just wanted to thank all of our speakers for today, and Srinivas, as well as all of our audience members for your questions as well. This ends the AgriSeq or Agrigenomics session for day 2. We will now be taking a 15-minute break before we start the sessions in other tracks. So you can have a coffee break and visit our booths, learn a bit more about some of the very exciting promotions that we have made available. And you can also ask Thermo Fisher representatives there any questions you may have as well. Please also participate in the scavenger hunting game. Look for our icons in the environment to win a number of prizes there. There will also be a pop-up survey appearing in a minute, so please complete the survey and it will help us plan for these events in future. Thank you again to our speakers, and I look forward to speaking to you all again in about 15 minutes. Thank you.